tribal member of the Confederate Tribes of the Umatilla Indian Reservation. Today, I will be talking about the biochemistry of a medicinal root that is used and known by our people as couch. This sacred root was used for thousands of years by people for the sacred and medicinal purpose it provides. The couch contains an organic compound known as terpenes. Terpenes are hydrocarbons which act as protective compounds found in other plants and insects. The majority of the terpene profile found in couch is composed of cymene, transaminal acetate, pyridine, mercerine, and p-cymene. We will focus on one of these, p-cymene, which has been demonstrated by Dr. Feng in Jilin University in China.
Nuclear factor kappa light chain enhancer of activated B cells, or NFKB, is a protein complex that controls transcription of DNA. This happens through a pathway within the cell when activated by stress stimuli such as cortisol, heavy metals, and cytokines. NFKB initiates an immune response to infection by increasing access to inflammatory gene upregulation. Overactive or unregulated NFKB activity may lead to health conditions including cancer and inflammatory diseases such as sepsis. However, p-cymene is able to prevent NFKB from accessing the DNA and producing unwanted inflammatory proteins, making couch a promising candidate for prevention and or treatment of NFK-related ailments. To explain how p-cymene functions as an anti-inflammatory agent, we will first need to consider how NFKB initiates an immune response within the body. When a stress factor activates a receptor such as toll-like 4 TLR4, a sequence that results in the phosphorylation of an enzyme complex IKB kinase, the enzyme transfers the phosphate to a protein complex known as IKBA, an inhibitor of NFKB, degrading it and allowing NFKB to enter the nucleus where it targets and binds pro-inflammation gene. NFKB is then joined by additional nuclear factors such as RNA polymerase, which generates messenger mRNA. Once complete, the mRNA exits the nucleus and is converted to an inflammatory response protein by the ribosome. There are several ways to prevent the inflammatory genes from being produced. While the current research has not identified the specific target of p-cymene, we can hypothesize that one of the pathways could be preventing inflammatory gene expression. We will consider 3 possible ways for p-cymene to prevent NFKB from initiating transcription of inflammatory genes: initial blocking of the stress factor receptor, phosphorylation prevention of IKB kinase, phosphorylation prevention of IKB. Each of these pathways can prevent NFKB from activating the inflammatory genes.
Millennia, our ancestors used the resources available to solve the health problems. Herbal medicine is used in a variety of our cultural practices, such as sweat lodges, sun dances, and during our powwows. This study is an example of confirming our spiritual understanding with our current scientific understanding. Thank you for listening. Katsuyaya.